My name is Shweta.
And my name is Bella, and today we're going to explain the process of protein synthesis. So, protein synthesis is the name of the process of building proteins, and it occurs in the ribosomes. So, the two major steps are transcription and translation. Transcription occurs when the transcript of DNA is generated into mRNA. So, during transcription, RNA polymerase binds to DNA and separates the two strands. It then uses one of the strands of the DNA as a template where nucleotides are attached into a string of RNA. So, base pairing rules apply as they would in DNA. Guanine will connect to cytosine. The only difference is that adenine will connect to uracil instead of thymine. So once the base pairs are connected, it will look something like this. Like I said, adenine will connect to uracil, cytosine will connect to guanine, and because the template strand has thymine already, it will connect to adenine. So the finishing product will look like this, but once everything is solidified and it comes together, the product will look like this. So this is after all of the base pairs connect, and yeah. So the next step is that the strand of RNA will move and connect to the surface of the ribosome. So codons are 3 pairs of bases. So this first codon is UUU, and so like I said, U bonds with A, so then the pair would be AAA. So that's the first. This is tRNA, and this is an amino acid. So the next bond would be UCG, that's the codon, and then the tRNA has A, G, and C. A connects with U, G connects with C, and C connects with G. So the final codon is UAA, and A bonds with U, U bonds with A, U bonds with A. So it'll look a little bit something like this. So these are the amino acids, and these are the tRNAs.
Part of each promoter or special sequence of DNA is the initiation site. The template strand is used to add codons onto in order to form a new RNA strand. mRNA sends instructions to build amino acids to the rest of the cell.
Elongation is when RNA polymerase unwinds DNA, like I had mentioned, when the 2 original strands of DNA unwind due to the RNA polymerase. The RNA polymerase reads the template of the DNA strand from 3' to 5'. So 3' to 5' is 3 to 5. RNA transcript is a copy of the RNA that came from the DNA. So the whole process that we explained, how the RNA built off of the DNA, that is the transcript, essentially. So termination is specified by a specific DNA-based sequence. So before mRNA can leave the nucleus, which the nucleus is where the original DNA strand was, it has to be processed. So that means that the introns, which are the non-coding regions, they have to be removed, and the exons, which are the coding regions, are spliced together, which means they're connected together.
A cap and tail are installed to protect mRNA in cytoplasm. Translation turns mRNA into protein. It occurs in the ribosomes. The ribosomes are large and small subunits. The A site binds with anticodon of charged RNA. The P site is more tRNA, adds amino acids to the growing chain. The E site is where tRNA sits before being released. Each tRNA holds a specific amino acid. Ribosomes read the mRNA. Start codon is AUG. Anticodon is a sequence of 3 nucleotides to the original codons in the mRNA. Amino acids are combined specific order to form a polypeptide chain. It's a chain of amino acids linked together.
The stop codon stops translation and releases polypeptides. The start codon is the initiation signal for translation, so that's basically like the go sign, the green light. And this is a complete process of protein synthesis. Thank you for watching.